Because if you remove parts of it, cells can't live. Consider that at the time that people realized that we have to get the information out of DNA, studying a single gene made a PhD thesis and took years. So if you wanted to start on a project, like let's say you were studying some aspect of cancer, it would take you a long time, like maybe 2 or 3 years, just to try to find a gene that's involved in the thing you're studying. And then you could do some functional studies on that particular gene and then do something. So you might spend 5 or 10 years of your life just trying to find a single gene. Because cells and living systems are just black boxes. We don't know what gene controls what. Yet sequencing technology existed. So why not sequence genomes? Well, the reason is because at the time this was being proposed, I was sequencing DNA. And it took me about 3 days of work with radioactivity, trying to label molecules, to get just 100 bases of sequence. That meant if I wanted to sequence the 3 billion base human genome, it would take 250,000 years. So one solution is, well, hire 250,000 people and it'll take a year. But if you consider like the space program, some people get really pissed off about the space program because they say it's going to take enormous resources to do something like, go to Mars. And if we were to use that money in another way, we could accomplish a lot. But this is an investment in infrastructure. And so if you set a goal and then provide funding resources, people will attack that goal and the technology will change, and it has changed, and such that today, using just off-the-shelf technology, we can collect a human genome, 3 billion bases in about 3 weeks. And that is changing on a daily basis, which means that, in the not too distant future, when you go to your doctor's office, you might know the sequence of your own genome, which can be really important for various things. Another way to look at this problem is if information content is a limiting aspect in biology. Around the time that genomes were starting to be sequenced was around here, and this is where we are now. So this is a graph of the information content in GenBank. So when you have an event like this, where you go from a long time with almost no information, and then suddenly a huge shift like this, things change. It causes a paradigm shift in the way that you think about the system that you're studying. This even looks a little bit like internet usage, where back here almost no one was on the internet, and now almost everybody is. So genomes again. Just because we have the sequence of a genome doesn't mean that we know what any of it codes for, right? We can try to predict where genes are, but we still don't know what those genes do. In addition, one of the fundamental components which makes like an ape or a worm different than a human is not just the genes, because we share so many genes in common with yeast and worms and apes, but the control elements that turn those genes on or off. This is a ripe area of biology which is waiting to be explored. It's called the cis-regulatory code. It's the control mechanisms for genes. So having all this information, primarily what it does is it causes people to think about the system in a new way. If you're studying a black box and you don't have a good map of it, you're going to sort of push on it in one place and see what happens in another place. But if you have an entire map, it's like suddenly having a map of the world and you get to now annotate it. Did you have a question? Is the information content of a genome both necessary and sufficient? It depends on the organism. For instance, so let me just talk about this for a second. Thinking about the whole system. In 1996, yeast was the first eukaryotic organism sequenced. Now you've got 12 megabases of sequence. You can predict that there's about 6,000 genes. For the first time, people are realizing, wait a minute, I need to know what a database is because I need to keep track of all these genes. There are methods for knocking genes out of yeast. So if you've got 6,000 targets and a lot of graduate students, you can say, well, let's start knocking out genes and see what happens. So what was done is that the yeast community made a knockout collection. They tried to make 6,000 different strains of yeast in which each yeast was missing a different gene. So of that, they were able to make about 4,000 strains because about 2,000 of the genes are absolutely essential. If you knock out the gene, the organism is dead, right? So necessary and sufficient, it sort of depends. Some single knockouts, you knock out gene X, the yeast are alive. You go to knock out a second gene, now the yeast are dead. Is there information content required for organisms that's outside the genome? What's that? Well, there's metagenomic elements. So, like, E. coli has a genome. Yeast have genomes. But they also have plasmids. And these plasmids are like little circles of DNA that can be shifted between strains. Well, mitochondrial DNA is also, yeah, so mitochondrial DNA is sort of an extra genomic element. Yeah, so, you know, what's an organism anyway? I mean, look at mad cow disease. You have a protein which is infectious. There's no nucleic acid. It's a structure. It's a self-propagating structure. So one thing that changed, along with all this, okay, yeast, you've got 6,000 genes, 6,000 targets. Well, make copies of all the genes. So do 6,000 PCR reactions and make 6,000 pieces of DNA, and then build a robot. I mean, most molecular biologists are not robot builders, but it's actually not hard to build these robots. They use off-the-shelf technology. So here's an XYZ robot. This is a linear stage. This is a linear stage. This little thing here's a linear stage. So here's a homemade robot that can execute motion in 3 dimensions. So people started realizing, well, if you can have an XYZ robot and you've got lots of genetic elements, there are a lot of things you can do. One thing that we do is we take and we create slides in which we spot down a little bit of every yeast gene on a slide. So here's a slide that's got 6,000 spots on it representing the entire yeast genome on a hunk of glass. So for the first time ever, remember I said that people used to study genes one at a time, right? Well, why do an experiment to measure just one gene if you can measure 6,000 genes? If you do measure 6,000 genes, this is sort of what the data looks like if you scan that slide after doing an experiment. Each one of these dots is a different gene. The color of the dot represents the level of expression of that gene. But you don't even have to know what the genes do to measure them. But let's say you're comparing, you know, healthy tissue to cancerous tissue, and you see some spots fluctuating. Those are your things to go study, right? And now you've found a bunch of genes. The difficulty here is that nobody knows what to do. This is a 6,000 point vector representing the state of a system. Again, most molecular biologists are not very good at math. That's why people go into biology, right? So how do you know, and this is every experiment you do is going to generate a 6,000 point vector with yeast. You do it in humans, you're going to have 40,000 point vectors. How do you compare a 40,000 point vector? How do you compare 100 different experiments? So the data generation is huge. You know, physicists and mathematicians are getting involved now. But the main point I want to make is that now you've gone from a black box that you used to be able to poke and measure one output, but now you can poke it and measure the entire state of the transcriptional system. You can measure you can get a snapshot of the activity of all the genes at once. And this is really powerful. And so this is, you've probably heard the term genomics. This is what genomics is. It's instead of thinking of things one at a time, you think of them everything at a time. If you want to study spinal cord development, study snakes. They're all spinal cord, right? But that means you have to sequence the genome. But we can sequence the genome now. So the whole genome of the organism. You want to measure all the genes? Do an array experiment. You can measure all the genes. But you know what I showed you was measuring the RNA level of a gene. That RNA has to be translated into protein, and that is also a regulated step. So there are methods for studying all the proteins of an organism at once. All these proteins interact with each other. Proteins interact with each other. They interact with DNA. There's all kinds of interactions in the cell. So if you want to think about trying to catalog those things, you can do high throughput screens to try to measure all the interactions at any given time. But again, it's just one point in time. You know, if you heat the cells, this happens. If you cool the cells down, this happens. If you expose the cells to aspirin, that happens. If you do something else, put them in UV light, whatever. So with all this information, now people are thinking in a different way. Everybody wants to have a network map of their organism. So this is a map of E. coli, and it's protein-protein interactions in E. coli, and you can start seeing structures from these maps. I mean, people have been studying networks for a long time, but we don't know really what they mean in biology. Here's another map of yeast protein-protein interactions. More proteins, more interactions.
What are the lines?
Yeah, so the dots are individual proteins, and the lines between the dots reflect that somebody has measured an interaction between those proteins. So one question is, I mean, if you're going to measure 6,000 things here and 6,000 things there and do, a year of experiments to measure all the protein-protein interactions, you have to have a way to summarize the data, to view the data, to do something with it. Most people I know, they want to be able to draw these pictures, but they don't know how to do any sort of network math. They don't know how to compute on these things. They don't necessarily know, does this thing have any predictive power? What does it tell me? How can I compute on this structure? Which is, I think that's a good thing to do, but even if you can't do that, you could probably look at this structure right here and realize that the protein at the center is probably a really important protein. But along with these sort of network maps, people also started thinking of their data in terms of these kinds of structures. So here's a little map summarizing sea urchin development. It turns out that this map is also true for humans, that sea urchins and humans early on have very similar developmental events. And you can sort of represent those events in terms of figures like this. And when I first saw this, the first thing I thought of was it looks like a circuit diagram. There's things which are activators, there's things which are repressors. And if you can encode your biological system with something like this, now I kind of want to know, how do I tweak this? How do I understand the different components? It's obviously composed of modules. It's got all of these different parts. Shouldn't we be able to sort of look at this big catalog of parts and make our own network diagram of something? Yeah? Can it be displayed as an algorithmic table? Yeah, I think it can. Yeah. So if we revisit now, what does biohacking mean? So where does all that just leave us? It leaves us now we're swimming with information. We're surrounded by genes and parts. We're surrounded by entire systems level snapshots of activity. So now people think about things in a different way. So if we want to now probe systems, we can start asking questions along this level. You want to understand the system, so you can sort of change existing systems. Now you can see the system, you can change it. You can knock out genes. You can study, remember there's genes and there's promoters which control the genes. So you can try to understand what the activity of the promoters is. You can put genes in places where they're not supposed to be. Maybe there's a gene in your toenail that's not supposed to be expressed in your eyeball. So what happens if you express a toenail gene in your eyeball? If you have genes, you can not only sort of misexpress them and knock them out, but you can try changing them to see what happens if you tweak them. You can try to create new proteins, which is almost impossible because try to start with just general sequence space and evolve something new, it's basically impossible. It's impossible because it takes too much DNA to try to search through any kind of sequence space. But it's also impossible because when you actually try to do it, you end up evolving things which bind to glass and plastic, and you don't get anywhere. But that doesn't mean that there aren't other ways to go about making new proteins. And these days, we also talk a lot about, can we just create whole new systems? Given that we've got so many views of these systems, can we just create new ones? So before I talk about some individual circuits, I wanted to just remind people of, or maybe introduce people to a few basic components. Here's a figure of a gene. It would be expressed in this direction. And remember that genes have promoters, and whether this gene gets expressed or not depends on the sequences in this promoter, and it depends on whether there's proteins around which will bind this promoter and turn the gene on, or other proteins which will bind this promoter and keep the gene off. Proteins which bind and keep the gene off are called repressors. And repressors can be inactivated. For instance, if you're a bacteria and you have a gene for lactose, you don't want to express that gene unless there's actually lactose around in your environment. So the lactose gene will be repressed until there's lactose around. And what lactose will do, it interacts with the protein, and when it interacts with the protein, the repressor comes off, and now the gene is expressed. So there's activators and there's repressors, and this is at least in bacteria. In mammalian systems, things are just way more complicated. But nonetheless, given a bunch of